Hi guys, I'm Angeline and today I'm going to talk about genetic engineering that can modify cows and make them produce less nutrients. So the reason why I chose this topic is because most of my mother's side relatives do not eat beefs, since they believe that cows helped them a lot in the past few decades under agriculture such as plowing and carrying all the merchandise. So they think we shouldn't eat beef as an exchange and appreciation to their efforts. So cows play an important role in our lives. But what's the main deal with cows? It's their fart that encourages scientists to study and genetic engineer them. Their fart is mainly composed of methane, which is a highly potent gas and it's really destructive to the environment. So first of all, why do cows fart methane? Every organism needs microorganisms to digest at certain degrees. During digestion, microbes in a cow's digestive system ferment the diets that include high cellulosic plants such as grass and hay. And this microbial fermentation process produces methane as a byproduct, which can be exhaled or belched by the cows. These microorganisms belong to the domain Archaea, and they use hydrogen gas and carbon dioxide to produce methane. This regulates fermentation in the rumen. For cows, those microbes are necessary and important to gain the nutrition and energy from their diet. Therefore, by modifying or genetic engineering the microbes, more specifically methanogens in the cow's rumen, can change the methane production. So now we are targeting the bacteria. As shown in the picture, this is what a normal bacteria looks like. Plasmids are used as vectors in genetic engineering. Aside from the bacterial DNA as shown in the picture, plasmid is a genetic structure in a cell that can replicate independently of the chromosomes. It's typically a small circular DNA strand in the cytoplasm of a bacterium, normally floating freely. These plasmids serve as important tools in genetics and biotechnology labs, because bacteria can naturally pick up plasmids from their environment, and this makes scientists easier to transfer their desired genetic trait. So now let's move on to the basic and general process of genetic engineering. So first of all, we have to extract the bacteria from the cow's rumen, and this is what the bacteria looks like. And then we have to take the plasmid out of the bacteria. Then by using the restriction enzyme, we can remove the unwanted part of the plasmid and turn it into a DNA segment that has sticky ends. The same works for the foreign DNA, which is what we will be adding to the plasmid. Same restriction enzyme is used to cut the foreign DNA and select the specific gene of interest. Both of the segments have sticky ends, and the ligase is used to combine the two together. And this results in the recombinant plasmid DNA. In a larger image, this is the selected plasmid, and while this is the selected interest of gene. Connected by the ligase, they result in the recombinant plasmid DNA. The recombinant plasmid DNA is inserted back to the bacteria. And now, the bacteria can produce less methane. Yay! There is still one last thing we have to make sure about the recombinant plasmid DNA. With only the genetic information coding for producing less methane, we couldn't distinguish whether the bacteria undergo transformation or not. Transformation is the process in which recombinant plasmid DNA are transcribed into proteins, and the desired trait is expressed. In order to identify whether or not the bacteria undergo transformation, we should add an antibiotic-resistant gene to the recombinant plasmid DNA using the same previous method, shown as the yellow part of the plasmid. The reason why we should add antibiotic resistant to the plasmid DNA is because that we can expose the bacteria into antibiotics. Once the bacteria undergoes transformation, like the one shown above, it is resistant to antibiotics. On the other hand, the bacteria that did not undergo transformation will be killed because of their lack of resistance. By using this method, we can efficiently distinguish between the untransformed bacteria and transformed bacteria, just like what we learned in class about ampicillin. This is similar to the process of gene cloning, which is what we learned in class. The only difference is that gene cloning is used to produce large quantities of the recombinant plasmid DNA. Now let's talk about how methane is a threat to Earth. Methane is a major contributor, second only to carbon dioxide to greenhouse effect. The level of destruction methane is bringing to the atmosphere is 23 times worse than carbon dioxide. To emphasize its destruction, compare it with a daily life object, the cars. The average amount of methane produced by 2 cows is equally detrimental to the average amount of carbon dioxide produced by a car annually. So what exactly is greenhouse effect? Let's draw an analogy here. Think of sun's radiation as the fire of the stove and the atmosphere as the frying pan, while the greenhouse gases are the ingredients on the pan. The ingredients on the pan will absorb the heat from the fire, and this works the same for greenhouse gases. The greenhouse gases trap the heat that passes through the atmosphere, then spread it to their surroundings and warming up the surface of Earth. This eventually leads to global warming, a current issue that affects everyone. Global warming reminds us to be aware and pay more attention on the environment in order to leave a better place for our future generations. To give you a better idea, these are some contributors to the greenhouse effect, and as I mentioned before, methane is a major one. Let's see how greenhouse effect is in relation with global warming. As the graph indicates, there is a correlation between the amount of methane in the atmosphere and the temperature change in the past history. Note that the progression of time in the graph is from present to past. Shown in the graph, methane, the main component of cow's fart, is the direct cause of global warming. Therefore, we can conclude that greenhouse effect is one of the reasons why global warming occurs. This is another graph similar to the previous one that also shows the relationship between global warming and greenhouse effect. Both carbon dioxide and methane are greenhouse gases, but they display different levels of influence to the environment. Let's get into global warming. Over the past decades, Earth had experienced the fastest rate of increasing temperatures. Some of the consequences of global warming include melting glaciers, early snow melt, and rising sea levels, which will lead to flooding in many areas. And the disruption of climate also may lead to death of certain organisms or extinction. There are just too many problems that global warming may lead to, so scientists are finding many ways to improve the environment and make the rate of global warming slower. And genetic engineering the bacteria in the cow's rumen might be one of the possible solutions. So now let's compare and contrast the pros and cons of the project. For the pros, I think there are the main reasons why we should genetic engineer those microorganisms. First, it can cut the methane emission up to 25%. And second, it can slow down the rate of global warming by decreasing the occurrence of greenhouse effects. However, for the cons, there are many concerns about ethical issues and the health of humans. So, some of the examples may be, humans may breed cows uncontrollably since they think that methane emission is no longer an issue. And second, the quality of cow's digestion might go down and affect the quality of beef, which may lead to problems in human health. Lastly, since humans may breed cows uncontrollably, the overconsumption of beef may also lead to obesity or other disease. Overall, I think the pros outweigh the cons since global warming is now an important issue, and we only have one Earth. We should save as much as possible for the future generations. By producing less methane, it sure would help the environment. Lastly, there are some acknowledgements that I have to make about the project. Due to scarce information of genetically engineering cows, the overall ideas presented in the video are general. In reality, instead of directly genetically engineer the cows, scientists work more upon the adding of supplementary nutrition to cows' diets and researching about the bacteria in their rumens, which is what I focused on in my project. So sorry guys for having such a misleading topic, and hope you guys enjoyed my video. Bye!